Introduction Chapter Objectives. After studying this chapter, you will be able to identify the main tissue types and discuss their roles in the human body. Identify the 4 types of tissue membranes and the characteristics of each that make them functional. Explain the functions of various epithelial tissues and how their forms enable their functions. Explain the functions of various connective tissues and how their forms enable their functions. Describe the characteristics of muscle tissue and how these enable function. Discuss the characteristics of nervous tissue and how these enable information processing and control of muscular and glandular activities. The body contains at least 200 distinct cell types. These cells contain essentially the same internal structures, yet they vary enormously in shape and function. The different types of cells are not randomly distributed throughout the body. Rather, they occur in organized layers, a level of organization referred to as tissue. The micrograph that opens this chapter shows the 135 high degree of organization among different types of cells in the tissue of the cervix. You can also see how that organization breaks down when cancer takes over the regular meiotic functioning of a cell. The variety in shape reflects the many different roles that cells fulfill in your body. The human body starts as a single cell at fertilization. As this fertilized egg divides, it gives rise to trillions of cells, each built from the same blueprint, but organizing into tissues and becoming irreversibly committed to a developmental pathway. 4.1 Types of tissues. By the end of this section, you will be able to identify the 4 main tissue types, discuss the functions of each tissue type, relate the structure of each tissue type to their function, discuss the embryonic origin of tissue, identify the 3 major germ layers, identify the main types of tissue membranes. The term tissue is used to describe a group of cells found together in the body. The cells within a tissue share a common embryonic origin. Microscopic observation reveals that the cells in a tissue share morphological features and are arranged in an orderly pattern that achieves the tissue's functions. From the evolutionary perspective, tissues appear in more complex organisms. For example, multicellular protists, ancient eukaryotes, do not have cells organized into tissues. Although there are many types of cells in the human body, they are organized into 4 broad categories of tissues: epithelial, connective, muscle, and nervous. Each of these categories is characterized by specific functions that contribute to the overall health and maintenance of the body. A disruption of the structure is a sign of injury or disease. Such changes can be detected through histology, the microscopic study of tissue appearance, organization, and function. The 4 types of tissues. Epithelial tissue, also referred to as epithelium, refers to the sheets of cells that cover exterior surfaces of the body, lines internal cavities and passageways, and forms certain glands. Connective tissue, as its name implies, binds the cells and organs of the body together and functions in the protection, support, and integration of all parts of the body. Muscle tissue is excitable, responding to stimulation and contracting to provide movement, and occurs as 3 major types: skeletal, voluntary, muscle, smooth muscle, and cardiac muscle in the heart. Nervous tissue is also excitable, allowing the propagation of electrochemical signals in the form of nerve impulses that communicate between different regions of the body. Figure 4.2. The next level of organization is the organ, where several types of tissues come together to form a working unit. Just as knowing the structure and function of cells helps you in your study of tissues, knowledge of tissues will help you understand how organs function. The epithelial and connective tissues are discussed in detail in this chapter. Muscle and nervous tissues will be discussed only briefly in this chapter. 136 1.8 Figure 4.24 Types of tissue. Body. The 4 types of tissues are exemplified in nervous tissue, stratified squamous epithelial tissue, cardiac muscle tissue, and connective tissue in small intestine. Clockwise from nervous tissue: LM times 872, LM times 282, LM times 460, LM times 800. Micrographs provided by the Regents of University of Michigan Medical School. Copyright 2012. Embryonic origin of tissues. The zygote, or fertilized egg, is a single cell formed by the fusion of an egg and sperm. After fertilization, the zygote gives rise to rapid meiotic cycles, generating many cells to form the embryo. The first embryonic cells generated have the ability to differentiate into any type of cell in the body and, as such, are called totipotent, meaning each has the capacity to divide, differentiate, and develop into a new organism. As cell proliferation progresses, three major cell lineages are established within the embryo. As explained in a later chapter, each of these lineages of embryonic cells forms the distinct germ layers from which all the tissues and organs of the human body eventually form. Each germ layer is identified by its relative position. Ectoderm, ecto equals outer, mesoderm, meso equals middle, and endoderm, endo equals inner. Figure 4.3 shows the types of tissues and organs associated with each of the three germ layers. Note that epithelial tissue originates in all three layers, whereas nervous tissue derives primarily from the ectoderm and muscle tissue from mesoderm. 137 Figure 4.3 Embryonic origin of tissues and major organs. View this slideshow. http://openstaxcollege.org/l/stem cells to learn more about stem cells. How do somatic stem cells differ from embryonic stem cells? 138 1.8 Tissue membranes. A tissue membrane is a thin layer or sheet of cells that covers the outside of the body, for example, skin, the organs, for example, pericardium, internal passageways that lead to the exterior of the body, for example, abdominal mesenteries, and the lining of the movable joint cavities. There are two basic types of tissue membranes: connective tissue and epithelial membranes. Figure 4.4. Figure 4.4 Tissue membranes. The two broad categories of tissue membranes in the body are 1. Connective tissue membranes, which include synovial membranes, and 2. Epithelial membranes, which include mucous membranes, serous membranes, and the cutaneous membrane, in other words, the skin. Connective tissue membranes. The connective tissue membrane is formed solely from connective tissue. These membranes encapsulate organs, such as the kidneys, and line our movable joints. A synovial membrane is a type of connective tissue membrane that lines the cavity of a freely movable joint. For example, synovial membranes surround the joints of the shoulder, elbow, and knee. Fibroblasts in the inner layer of the synovial membrane release hyaluronan into the joint cavity. The hyaluronan effectively traps available water to form the synovial fluid, a natural lubricant that enables the bones of a joint to move freely against one another without much friction. This synovial fluid readily exchanges water and nutrients with blood, as do all body fluids. Epithelial membranes. The epithelial membrane is composed of epithelium attached to a layer of connective tissue, for example, your skin. The mucous membrane is also a composite of connective and epithelial tissues, sometimes called mucosae. These epithelial membranes line the body cavities and hollow passageways that open to the external environment, and include the digestive, respiratory, excretory, and reproductive tracts. Mucus, produced by the epithelial exocrine glands, covers the epithelial layer. The underlying connective tissue, called the lamina propria, literally, own layer, helps support the fragile epithelial layer. A serous membrane is an epithelial membrane composed of mesodermally derived epithelium called the mesothelium that is supported by connective tissue. These membranes line the coulommic cavities of the body, that is, those cavities that do not open to the outside, and they cover the organs located within those cavities. They are essentially membranous bags, with mesothelium lining the inside and connective tissue on the outside. Serous fluid secreted by the cells of the thin squamous mesothelium lubricates the membrane and reduces abrasion and friction between organs. Serous membranes are identified according locations. 3 serous membranes line the thoracic cavity, the 2 pleura that cover the lungs and the pericardium that covers the heart. A fourth, the peritoneum, is the serous membrane in the abdominal cavity that covers abdominal organs and forms double sheets of mesenteries that suspend many of the digestive organs. The skin is an epithelial membrane, also called the cutaneous membrane. It is a stratified squamous epithelial membrane resting on top of connective tissue. The apical surface of this membrane is exposed to the external environment and is covered with dead, keratinized cells that help protect the body from desiccation and pathogens. 4.2. Epithelial tissue. By the end of this section, you will be able to explain the structure and function of epithelial tissue, distinguish between tight junctions, anchoring junctions, and gap junctions, distinguish between simple epithelia and stratified epithelia, as well as between squamous, cuboidal, and columnar epithelia. Describe the structure and function of endocrine and exocrine glands and their respective secretions. Most epithelial tissues are essentially large sheets of cells covering all the surfaces of the body, exposed to the outside world and lining the outside of organs. Epithelium also forms much of the glandular tissue of the body. Skin is not the only area of the body exposed to the outside. Other areas include the airways, the digestive tract, as well as the urinary and reproductive systems, all of which are lined by an epithelium. Hollow organs and body cavities that do not connect to the exterior of the body, which includes blood vessels and serous membranes, are lined by endothelium, plural equals endothelia, which is a type of epithelium. Epithelial cells derive from all three major embryonic layers: the epithelia lining the skin, parts of the mouth and nose, and the anus develop from the ectoderm. Cells lining the airways and most of the digestive system originate in the endoderm. The epithelium that lines vessels in the lymphatic and cardiovascular system derives from the mesoderm and is called an endothelium. All epithelia share some important structural and functional features. This tissue is highly cellular, with little or no extracellular material present between cells. Adjoining cells form a specialized intercellular connection between their cell membranes called a cell junction. The epithelial cells exhibit polarity with differences in structure and function between the exposed or apical-facing surface of the cell and the basal surface close to the underlying body structures. The basal lamina, a mixture of glycoproteins and collagen, provides an attachment site for the epithelium, separating it from underlying connective tissue. The basal lamina attaches to a reticular lamina, which is secreted by the underlying connective tissue, forming a basement membrane that helps hold it all together. Epithelial tissues are nearly completely avascular. For instance, no blood vessels cross the basement membrane to enter the tissue, and nutrients must come by diffusion or absorption from underlying tissues or the surface. Many epithelial tissues are capable of rapidly replacing damaged and dead cells. Sloughing off of damaged or dead cells is a characteristic of surface epithelium and allows our airways and digestive tracts to rapidly replace damaged cells with new cells. 1.8 Generalized functions of epithelial tissue. Epithelial tissues provide the body's first line of protection from physical, chemical, and biological wear and tear. The cells of an epithelium act as gatekeepers of the body, controlling permeability and allowing selective transfer of materials across a physical barrier. All substances that enter the body must cross an epithelium. Some epithelia often include structural features that allow the selective transport of molecules and ions across their cell membranes. Many epithelial cells are capable of secretion and release mucus and specific chemical compounds onto their apical surfaces. The epithelium of the small intestine releases digestive enzymes, for example. Cells lining the respiratory tract secrete mucus that traps incoming microorganisms and particles. A glandular epithelium contains many secretory cells. The epithelial cells are typically characterized by the polarized distribution of organelles and membrane-bound proteins between their basal and apical surfaces. Particular structures found in some epithelial cells are an adaptation to specific functions. Certain organelles are segregated to the basal sides, whereas other organelles and extensions, such as cilia, when present, are on the apical surface. Cilia are microscopic extensions of the apical cell membrane that are supported by microtubules. They beat in unison and move fluids as well as trapped particles. Ciliated epithelium lines the ventricles of the brain where it helps circulate the cerebrospinal fluid. The ciliated epithelium of your airway forms a mucociliary escalator that sweeps particles of dust and pathogens trapped in the secreted mucus toward the throat. It is called an escalator because it continuously pushes mucus with trapped particles upward. In contrast, nasal cilia sweep the mucus blanket down towards your throat. In both cases, the transported materials are usually swallowed, and end up in the acidic environment of your stomach. Cell-to-cell junction cells of epithelia are closely connected and are not separated by intracellular material. Three basic types of connections allow varying degrees of interaction between the cells: tight junctions, anchoring junctions, and gap junctions. Figure 4.5. Figure 4.5 Types of cell junctions. The three basic types of cell-to-cell junctions are tight junctions, gap junctions, and anchoring junctions. At one end of the spectrum is the tight junction, which separates the cells into apical and basal compartments. When two adjacent epithelial cells form a tight junction, there is no extracellular space between them and the movement of substances through the extracellular space between the cells is blocked. This enables the epithelia to act as selective barriers. An anchoring junction includes several types of cell junctions that help stabilize epithelial tissues. Anchoring junctions are common on the lateral and basal surfaces of cells where they provide strong and flexible connections. There are three types of anchoring junctions: desmosomes, hemidesmosomes, and adherens. Desmosomes occur in patches on the membranes of cells. The patches are structural proteins on the inner surface of the cell's membrane. The adhesion molecule, cadherin, is embedded in these patches and projects through the cell membrane to link with the cadherin molecules of adjacent cells. These connections are especially important in holding cells together. Hemidesmosomes, which look like half a desmosome, link cells to the extracellular matrix, for example, the basal lamina. While similar in appearance to desmosomes, they include the adhesion proteins called integrins rather than cadherins. Adherens junctions use either cadherins or integrins depending on whether they are linking to other cells or matrix. The junctions are characterized by the presence of the contractile protein actin located on the cytoplasmic surface of the cell membrane. The actin can connect isolated patches or form a belt-like structure inside the cell. These junctions influence the shape and folding of the epithelial tissue. In contrast with the tight and anchoring junctions, a gap junction forms an intercellular passageway between the 1.8 membranes of adjacent cells to facilitate the movement of small molecules and ions between the cytoplasm of adjacent cells. These junctions allow electrical and metabolic coupling of adjacent cells, which coordinates function in large groups of cells. Classification of epithelial tissues. Epithelial tissues are classified according to the shape of the cells and number of the cell layers formed. Figure 4.6. Cell shapes can be squamous, flattened and thin, cuboidal, boxy, as wide as it is tall, or columnar, rectangular, taller than it is wide. Similarly, the number of cell layers in the tissue can be 1, where every cell rests on the basal lamina, which is a simple epithelium, or more than 1, which is a stratified epithelium and only the basal layer of cells rests on the basal lamina. Pseudostratified. Pseudo equals false. Describes tissue with a single layer of irregularly shaped cells that give the appearance of more than one layer. Transitional describes a form of specialized stratified epithelium in which the shape of the cells can vary. Figure 4.6 Cells of epithelial tissue. Simple epithelial tissue is organized as a single layer of cells and stratified epithelial tissue is formed by several layers of cells. Simple epithelium. The shape of the cells in the single-cell layer of simple epithelium reflects the functioning of those cells. The cells in simple squamous epithelium have the appearance of thin scales. Squamous cell nuclei tend to be flat, horizontal, and elliptical, mirroring the form of the cell. The endothelium is the epithelial tissue that lines vessels of the lymphatic and cardiovascular system, and it is made up of a single layer of squamous cells. Simple squamous epithelium, because of the thinness of the cell, is present where rapid passage of chemical compounds is observed. The alveoli of lungs where gases diffuse, segments of kidney tubules, and the lining of capillaries are also made of simple squamous epithelial tissue. The mesothelium is a simple squamous epithelium that forms the surface layer of the serous membrane that lines body cavities and internal organs. Its primary function is to provide a smooth and protective surface. Mesothelial cells are squamous epithelial cells that secrete a fluid that lubricates the mesothelium. In simple cuboidal epithelium, the nucleus of the box-like cells appears round and is generally located near the center of the cell. These epithelia are active in the secretion and absorptions of molecules. Simple cuboidal epithelia are observed in the lining of the kidney tubules and in the ducts of glands. In simple columnar epithelium, the nucleus of the tall column-like cells tends to be elongated and located in the basal end of the cells. Like the cuboidal epithelia, this epithelium is active in the absorption and secretion of molecules. Simple columnar epithelium forms the lining of some sections of the digestive system and parts of the female reproductive tract. Ciliated columnar epithelium is composed of simple columnar epithelial cells with cilia on their apical surfaces. These epithelial cells are found in the lining of the fallopian tubes and parts of the respiratory system, where the beating of the cilia helps remove particulate matter. Pseudostratified columnar epithelium is a type of epithelium that appears to be stratified but instead consists of a single layer of irregularly shaped and differently sized columnar cells. In pseudostratified epithelium, nuclei of neighboring cells appear at different levels rather than clustered in the basal end. The arrangement gives the appearance of stratification, but in fact all the cells are in contact with the basal lamina, although some do not reach the apical surface. Part 9. Pseudostratified columnar epithelium is found in the respiratory tract, where some of these cells have cilia. Both simple and pseudostratified columnar epithelia are heterogeneous epithelia because they include additional types of cells interspersed among the epithelial cells. For example, a goblet cell is a mucus-secreting unicellular gland, interspersed between the columnar epithelial cells of mucous membranes. Figure 4.7. A, B. Figure 4.7 Goblet cell, A. In the lining of the small intestine, columnar epithelium cells are interspersed with goblet cells. B. The arrows in this micrograph point to the mucus-secreting goblet cells. LM times 1600. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. 1.8 View the University of Michigan webscope at http://virtualslides.med.umich.edu/histology/digestive%20system/intestine/histo_histo_40x.svs/view.apml http://openstaxcollege.org/l/goblet to explore the tissue sample in greater detail. Stratified epithelium. A stratified epithelium consists of several stacked layers of cells. This epithelium protects against physical and chemical wear and tear. The stratified epithelium is named by the shape of the most apical layer of cells, closest to the free space. Stratified squamous epithelium is the most common type of stratified epithelium in the human body. The apical cells are squamous, whereas the basal layer contains either columnar or cuboidal cells. The top layer may be covered with dead cells filled with keratin. Mammalian skin is an example of this dry, keratinized, stratified squamous epithelium. The lining of the mouth cavity is an example of an unkeratinized, stratified squamous epithelium. Stratified cuboidal epithelium and stratified columnar epithelium can also be found in certain glands and ducts, but are uncommon in the human body. Another kind of stratified epithelium is transitional epithelium, so called because of the gradual changes in the shapes of the apical cells as the bladder fills with urine. It is found only in the urinary system, specifically the ureters and urinary bladder. When the bladder is empty, this epithelium is convoluted and has cuboidal apical cells with convex, umbrella-shaped, apical surfaces. As the bladder fills with urine, this epithelium loses its convolutions and the apical cells transition from cuboidal to squamous. It appears thicker and more multi-layered when the bladder is empty, and more stretched out and less stratified when the bladder is full and distended. Figure 4.8 summarizes the different categories of epithelial cell tissue cells. 145 Figure 4.8 Summary of epithelial tissue cells. 146 1.8 Watch this video. http://openstaxcollege.org/l/etissues to find out more about the anatomy of epithelial tissues. Where in the body would one find non-keratinizing stratified squamous epithelium? Glandular epithelium. A gland is a structure made up of one or more cells modified to synthesize and secrete chemical substances. Most glands consist of groups of epithelial cells. A gland can be classified as an endocrine gland, a ductless gland that releases secretions directly into surrounding tissues and fluids, endo equals inside, or an exocrine gland whose secretions leave through a duct that opens directly, or indirectly, to the external environment, exo equals outside. Endocrine glands. The secretions of endocrine glands are called hormones. Hormones are released into the interstitial fluid, diffused into the bloodstream, and delivered to targets, in other words, cells that have receptors to bind the hormones. The endocrine system is part of a major regulatory system coordinating the regulation and integration of body responses. A few examples of endocrine glands include the anterior pituitary, thymus, adrenal cortex, and gonads. Exocrine glands. Exocrine glands release their contents through a duct that leads to the epithelial surface. Mucus, sweat, saliva, and breast milk are all examples of secretions from exocrine glands. They are all discharged through tubular ducts. Secretions into the lumen of the gastrointestinal tract, technically outside of the body, are of the exocrine category. Glandular structure. Exocrine glands are classified as either unicellular or multicellular. The unicellular glands are scattered single cells, such as goblet cells, found in the mucous membranes of the small and large intestine. The multicellular exocrine glands, known as serous glands, develop from simple epithelium to form a secretory surface that secretes directly into an inner cavity. These glands line the internal cavities of the abdomen and chest and release their secretions directly into the cavities. Other multicellular exocrine glands release their contents through a tubular duct. The duct is single in a simple gland, but in compound glands is divided into one or more branches. Figure 4.9. In tubular glands, the ducts can be straight or coiled, whereas tubes that form pockets are alveolar, acinar, such as the exocrine portion of the pancreas. Combinations of tubes and pockets are known as tubulo-alveolar, tubulo-acinar, compound glands. In a branched gland, a duct is connected to more than one secretory group of cells. 147 Figure 4.9 Types of exocrine glands. Exocrine glands are classified by their structure. Methods and types of secretion. Exocrine glands can be classified by their mode of secretion and the nature of the substances released, as well as by the structure of the glands and shape of ducts. Figure 4.10. Merocrine secretion is the most common type of exocrine secretion. The secretions are enclosed in vesicles that move to the apical surface of the cell where the contents are released by exocytosis. For example, watery mucus containing the glycoprotein mucin, a lubricant that offers some pathogen protection, is a merocrine secretion. The eccrine glands that produce and secrete sweat are another example. 148 1.8 Figure 4.10 Modes of glandular secretion. In merocrine secretion, the cell remains intact. B. In apocrine secretion, the apical portion of the cell is released, as well. C. In holocrine secretion, the cell is destroyed as it releases its product and the cell itself becomes part of the secretion. Apocrine secretion accumulates near the apical portion of the cell. That portion of the cell and its secretory contents pinch off from the cell and are released. Apocrine sweat glands in the axillary and genital areas release fatty secretions that local bacteria break down. This causes body odor. Both merocrine and apocrine glands continue to produce and secrete their contents with little damage caused to the cell because the nucleus and Golgi regions remain intact after secretion. In contrast, the process of holocrine secretion involves the rupture and destruction of the entire gland cell. The cell accumulates its secretory products and releases them only when it bursts. New gland cells differentiate from cells in the surrounding tissue to replace those lost by secretion. The sebaceous glands that produce the oils on the skin and hair are holocrine glands, cells. Figure 4.11. 149 Figure 4.11 Sebaceous glands. These glands secrete oils that lubricate and protect the skin. They are holocrine glands and they are destroyed after releasing their contents. New glandular cells form to replace the cells that are lost. LM times 400. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. Glands are also named after the products they produce. The serous gland produces watery, blood plasma-like secretions rich in enzymes such as alpha amylase, whereas the mucus gland releases watery to viscous products rich in the glycoprotein mucin. Both serous and mucus glands are common in the salivary glands of the mouth. Mixed exocrine glands contain both serous and mucus glands and release both types of secretions. 4.3. Connective tissue supports and protects. By the end of this section, you will be able to identify and distinguish between the types of connective tissue: proper, supportive, and fluid. Explain the functions of connective tissues as may be obvious from its name. One of the major functions of connective tissue is to connect tissues and organs. Unlike epithelial tissue, which is composed of cells closely packed with little or no extracellular space in between, connective tissue cells are dispersed in a matrix. The matrix usually includes a large amount of extracellular material produced by the connective tissue cells that are embedded within it. The matrix plays a major role in the functioning of this tissue. The major component of the matrix is a ground substance often crisscrossed by protein fibers. This ground substance is usually a fluid, but it can also be mineralized and solid, as in bones. Connective tissues come in a vast variety of forms, yet they typically have in common three characteristic components: cells, large amounts of amorphous ground substance, and protein fibers. The amount and structure of each component correlates with the function of the tissue, from the rigid ground substance in bones supporting the body to the inclusion of specialized cells. For example, a phagocytic cell that engulfs pathogens and also rids tissue of cellular debris. Functions of connective tissues. Connective tissues perform many functions in the body, but most importantly, they support and connect other tissues. From the connective tissue sheath that surrounds muscle cells, to the tendons that attach muscles to bones, and to the skeleton that supports the positions of the body. Protection is another major function of connective tissue, in the form of fibrous capsules and bones that protect delicate organs and, of course, the skeletal system. Specialized cells in connective tissue defend the body from microorganisms that enter the body. Transport of fluid, nutrients, waste, and chemical messengers is ensured by specialized fluid connective tissues, such as blood and lymph. Adipose cells store surplus energy in the form of fat and contribute to the thermal insulation of the body. 151.8 Embryonic connective tissue. All connective tissues derive from the mesodermal layer of the embryo. See Figure 4.3. The first connective tissue to develop in the embryo is mesenchyme, the stem cell line from which all connective tissues are later derived. Clusters of mesenchymal cells are scattered throughout adult tissue and supply the cells needed for replacement and repair after a connective tissue injury. A second type of embryonic connective tissue forms in the umbilical cord, called mucous connective tissue or Wharton's jelly. This tissue is no longer present after birth, leaving only scattered mesenchymal cells throughout the body. Classification of connective tissues. The three broad categories of connective tissue are classified according to the characteristics of their ground substance and the types of fibers found within the matrix. Table 4.1. Connective tissue proper includes loose connective tissue and dense connective tissue. Both tissues have a variety of cell types and protein fibers suspended in a viscous ground substance. Dense connective tissue is reinforced by bundles of fibers that provide tensile strength, elasticity, and protection. In loose connective tissue, the fibers are loosely organized, leaving large spaces in between. Supportive connective tissue, bone and cartilage, provide structure and strength to the body and protect soft tissues. A few distinct cell types and densely packed fibers in a matrix characterize these tissues. In bone, the matrix is rigid and described as calcified because of the deposited calcium salts. In fluid connective tissue, in other words, lymph and blood, various specialized cells circulate in a watery fluid containing salts, nutrients, and dissolved proteins. Connective tissue examples: connective tissue proper, supportive connective tissue, fluid connective tissue, loose connective tissue, areolar adipose, reticular cartilage, hyaline fibrocartilage, elastic blood, dense connective tissue, regular elastic, irregular elastic, bones, compact bone, cancellous bone, lymph. Table 4.1 Connective tissue proper. Fibroblasts are present in all connective tissue proper. Figure 4.12. Fibrocytes, adipocytes, and mesenchymal cells are fixed cells, which means they remain within the connective tissue. Other cells move in and out of the connective tissue in response to chemical signals. Macrophages, mast cells, lymphocytes, plasma cells, and phagocytic cells are found in connective tissue proper but are actually part of the immune system protecting the body. 151 Figure 4.12 Connective tissue proper. Fibroblasts produce this fibrous tissue. Connective tissue proper includes the fixed cells fibrocytes, adipocytes, and mesenchymal cells. LM × 400. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. Cell types. The most abundant cell in connective tissue proper is the fibroblast. Polysaccharides and proteins secreted by fibroblasts combine with extracellular fluids to produce a viscous ground substance that, with embedded fibrous proteins, forms the extracellular matrix. As you might expect, a fibrocyte, a less active form of fibroblast, is the second most common cell type in connective tissue proper. Adipocytes are cells that store lipids as droplets that fill most of the cytoplasm. There are two basic types of adipocytes: white and brown. The brown adipocytes store lipids as many droplets, and have high metabolic activity. In contrast, white fat adipocytes store lipids as a single large drop and are metabolically less active. Their effectiveness at storing large amounts of fat is witnessed in obese individuals. The number and type of adipocytes depends on the tissue and location, and vary among individuals in the population. The mesenchymal cell is a multipotent adult stem cell. These cells can differentiate into any type of connective tissue cells needed for repair and healing of damaged tissue. The macrophage cell is a large cell derived from a monocyte, a type of blood cell, which enters the connective tissue matrix from the blood vessels. The macrophage cells are an essential component of the immune system, which is the body's defense against potential pathogens and degraded host cells. When stimulated, macrophages release cytokines, small proteins that act as chemical messengers. Cytokines recruit other cells of the immune system to infected sites and stimulate their activities. Roaming, or free, macrophages move rapidly by amoeboid movement, engulfing infectious agents and cellular debris. In contrast, fixed macrophages are permanent residents of their tissues. The mast cell, found in connective tissue proper, has many cytoplasmic granules. These granules contain the chemical signals histamine and heparin. When irritated or damaged, mast cells release histamine, an inflammatory mediator, which causes vasodilation and increased blood flow at a site of injury or infection, along with itching, swelling, and redness you recognize as an allergic response. Like blood cells, mast cells are derived from hematopoietic stem cells and are part of the immune system. Connective tissue fibers and ground substance. Three main types of fibers are secreted by fibroblasts: collagen fibers, elastic fibers, and reticular fibers. Collagen fiber is made from fibrous protein subunits linked together to form a long and straight fiber. Collagen fibers, while flexible, have great tensile strength, resist stretching, and give ligaments and tendons their characteristic resilience and strength. These fibers hold connective tissues together, even during the movement of the body. Elastic fiber contains the protein elastin along with lesser amounts of other proteins and glycoproteins. The main property of elastin is that after being stretched or compressed, it will return to its original shape. Elastic fibers are prominent in elastic tissues found in skin and the elastic ligaments of the vertebral column. Reticular fiber is also formed from the same protein subunits as collagen fibers. However, these fibers remain narrow and are arrayed in a branching network. They are found throughout the body, but are most abundant in the reticular tissue of soft organs, such as liver and spleen, where they anchor and provide structural support to the parenchyma, the functional cells, blood vessels, and nerves of the organ. All of these fiber types are embedded in ground substance. Secreted by fibroblasts, ground substance is made of polysaccharides, specifically hyaluronic acid, and proteins. These combine to form a proteoglycan with a protein core and 152 1.8 polysaccharide branches. The proteoglycan attracts and traps available moisture, forming the clear, viscous, colorless matrix you now know as ground substance. Loose connective tissue. Loose connective tissue is found between many organs where it acts both to absorb shock and bind tissues together. It allows water, salts, and various nutrients to diffuse through to adjacent or embedded cells and tissues. Adipose tissue consists mostly of fat storage cells, with little extracellular matrix, figure 4.13. A large number of capillaries allow rapid storage and mobilization of lipid molecules. White adipose tissue is most abundant. It can appear yellow and owes its color to keratin and related pigments from plant food. White fat contributes mostly to lipid storage and can serve as insulation from cold temperatures and mechanical injuries. White adipose tissue can be found protecting the kidneys and cushioning the back of the eye. Brown adipose tissue is more common in infants, hence the term, baby fat. In adults, there is a reduced amount of brown fat and it is found mainly in the neck and clavicular regions of the body. The mini mitochondria in the cytoplasm of brown adipose tissue help explain its efficiency at metabolizing stored fat. Brown adipose tissue is thermogenic, meaning that as it breaks down fats, it releases metabolic heat, rather than producing adenosine triphosphate, ATP, a key molecule used in metabolism. Figure 4.13 Adipose tissue. This is a loose connective tissue that consists of fat cells with little extracellular matrix. It stores fat for energy and provides insulation. LM × 800. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. Areolar tissue shows little specialization. It contains all the cell types and fibers previously described and is distributed in a random, web-like fashion. It fills the spaces between muscle fibers, surrounds blood and lymph vessels, and supports organs in the abdominal cavity. Areolar tissue underlies most epithelia and represents the connective tissue component of epithelial membranes, which are described further in a later section. Reticular tissue is a mesh-like, supportive framework for soft organs such as lymphatic tissue, the spleen, and the liver. Figure 4.14. Reticular cells produce the reticular fibers that form the network onto which other cells attach. It derives its name from the Latin reticulus, which means little net. 153 Figure 4.14 Reticular tissue. This is a loose connective tissue made up of a network of reticular fibers that provides a supportive framework for soft organs. LM × 1600. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. Dense connective tissue. Dense connective tissue contains more collagen fibers than does loose connective tissue. As a consequence, it displays greater resistance to stretching. There are two major categories of dense connective tissue: regular and irregular. Dense regular connective tissue fibers are parallel to each other, enhancing tensile strength and resistance to stretching in the direction of the fiber orientations. Ligaments and tendons are made of dense regular connective tissue, but in ligaments not all fibers are parallel. Dense regular elastic tissue contains elastin fibers in addition to collagen fibers, which allows the ligament to return to its original length after stretching. The ligaments in the vocal folds and between the vertebrae in the vertebral column are elastic. In dense irregular connective tissue, the direction of fibers is random. This arrangement gives the tissue greater strength in all directions and less strength in one particular direction. In some tissues, fibers crisscross and form a mesh. In other tissues, stretching in several directions is achieved by alternating layers where fibers run in the same orientation in each layer, and it is the layers themselves that are stacked at an angle. The dermis of the skin is an example of dense irregular connective tissue rich in collagen fibers. Dense irregular elastic tissues give arterial walls the strength and the ability to regain original shape after stretching. Figure 4.15. 154 1.8 Figure 4.15 Dense connective tissue. A dense regular connective tissue consists of collagenous fibers packed into parallel bundles. B. Dense irregular connective tissue consists of collagenous fibers interwoven into a mesh-like network. From top, Lm times 1000, Lm times 200. Micrographs provided by the Regents of University of Michigan Medical School. Copyright 2012. 155 Connective tissue. Tendinitis. Your opponent stands ready as you prepare to hit the serve, but you are confident that you will smash the ball past your opponent. As you toss the ball high in the air, a burning pain shoots across your wrist and you drop the tennis racket. That dull ache in the wrist that you ignored through the summer is now an unbearable pain. The game is over for now. After examining your swollen wrist, the doctor in the emergency room announces that you have developed wrist tendinitis. She recommends icing the tender area, taking non-steroidal anti-inflammatory medication to ease the pain and to reduce swelling, and complete rest for a few weeks. She interrupts your protests that you cannot stop playing. She issues a stern warning about the risk of aggravating the condition and the possibility of surgery. She consoles you by mentioning that well-known tennis players such as Venus and Serena Williams and Rafael Nadal have also suffered from tendinitis-related injuries. What is tendinitis and how did it happen? Tendinitis is the inflammation of a tendon, the thick band of fibrous connective tissue that attaches a muscle to a bone. The condition causes pain and tenderness in the area around a joint. On rare occasions, a sudden serious injury will cause tendinitis. Most often, the condition results from repetitive motions over time that strain the tendons needed to perform the tasks. Persons whose jobs and hobbies involve performing the same movements over and over again are often at the greatest risk of tendinitis. You hear of tennis and golfer's elbow, jumper's knee, and swimmer's shoulder. In all cases, overuse of the joint causes a microtrauma that initiates the inflammatory response. Tendinitis is routinely diagnosed through a clinical examination. In case of severe pain, X-rays can be examined to rule out the possibility of a bone injury. Severe cases of tendinitis can even tear loose a tendon. Surgical repair of a tendon is painful. Connective tissue in the tendon does not have abundant blood supply and heals slowly. While older adults are at risk for tendinitis because the elasticity of tendon tissue decreases with age, active people of all ages can develop tendinitis. Young athletes, dancers, and computer operators. Anyone who performs the same movements constantly is at risk for tendinitis. Although repetitive motions are unavoidable in many activities and may lead to tendinitis, precautions can be taken that can lessen the probability of developing tendinitis. For active individuals, stretches before exercising and cross-training or changing exercises are recommended. For the passionate athlete, it may be time to take some lessons to improve technique. All of the preventive measures aim to increase the strength of the tendon and decrease the stress put on it. With proper rest and managed care, you will be back on the court to hit that slice spin serve over the net. Watch this animation. http://openstaxcollege.org/l/tendinitis to learn more about tendinitis, a painful condition caused by swollen or injured tendons. Supportive connective tissues. Two major forms of supportive connective tissue, cartilage and bone, allow the body to maintain its posture and protect internal organs. 156 1.8 Cartilage. The distinctive appearance of cartilage is due to polysaccharides called chondroitin sulfates, which bind with ground substance proteins to form proteoglycans. Embedded within the cartilage matrix are chondrocytes, or cartilage cells, and the space they occupy are called lacunae, singular equals lacuna. A layer of dense, irregular connective tissue, the perichondrium, encapsulates the cartilage. Cartilaginous tissue is avascular, thus all nutrients need to diffuse through the matrix to reach the chondrocytes. This is a factor contributing to the very slow healing of cartilaginous tissues. The three main types of cartilage tissue are hyaline cartilage, fibrocartilage, and elastic cartilage. Figure 4.16. Hyaline cartilage, the most common type of cartilage in the body, consists of short and dispersed collagen fibers and contains large amounts of proteoglycans. Under the microscope, tissue samples appear clear. The surface of hyaline cartilage is smooth, both strong and flexible. It is found in the rib cage and nose and covers bones where they meet to form movable joints. It makes up a template of the embryonic skeleton before bone formation. A plate of hyaline cartilage at the ends of bone allows continued growth until adulthood. Fibrocartilage is tough because it has thick bundles of collagen fibers dispersed through its matrix. Menisci in the knee joint and the intervertebral discs are examples of fibrocartilage. Elastic cartilage contains elastic fibers as well as collagen and proteoglycans. This tissue gives rigid support as well as elasticity. Tug gently at your earlobes, and notice that the lobes return to their initial shape. The external ear contains elastic cartilage. 157 Figure 4.16 Types of cartilage. Cartilage is a connective tissue consisting of collagenous fibers embedded in a firm matrix of chondroitin sulfates. A. Hyaline cartilage provides support with some flexibility. The example is from dog tissue. B. Fibrocartilage provides some compressibility and can absorb pressure. C. Elastic cartilage provides firm but elastic support. From top, Lm times 300, Lm times 1200, Lm times 1016. Micrographs provided by the Regents of University of Michigan Medical School. Copyright 2012. Bone is the hardest connective tissue. It provides protection to internal organs and supports the body. Bone's rigid extracellular matrix contains mostly collagen fibers embedded in a mineralized ground substance containing hydroxyapatite, a form of calcium phosphate. Both components of the matrix, organic and inorganic, contribute to the unusual properties of bone. Without collagen, bones would be brittle and shatter easily. Without mineral crystals, bones would flex and provide little support. Osteocytes, bone cells like chondrocytes, are located within lacunae. The histology of transverse tissue from long bone shows a typical arrangement of osteocytes in concentric circles around a central canal. Bone is a highly vascularized tissue. Unlike cartilage, bone tissue can recover from injuries in a relatively short time. Cancellous bone looks like a sponge under the microscope and contains empty spaces between trabeculae, or arches of bone proper. It is lighter than compact bone and found in the interior of some bones and at the end of long bones. Compact bone is solid and has greater structural strength. 158 1.8 Fluid connective tissue. Blood and lymph are fluid connective tissues. Cells circulate in a liquid extracellular matrix. The formed elements circulating in blood are all derived from hematopoietic stem cells located in bone marrow. Figure 4.17. Erythrocytes, red blood cells, transport oxygen and some carbon dioxide. Leukocytes, white blood cells, are responsible for defending against potentially harmful microorganisms or molecules. Platelets are cell fragments involved in blood clotting. Some white blood cells have the ability to cross the endothelial layer that lines blood vessels and enter adjacent tissues. Nutrients, salts, and wastes are dissolved in the liquid matrix and transported through the body. Lymph contains a liquid matrix and white blood cells. Lymphatic capillaries are extremely permeable, allowing larger molecules and excess fluid from interstitial spaces to enter the lymphatic vessels. Lymph drains into blood vessels, delivering molecules to the blood that could not otherwise directly enter the bloodstream. In this way, specialized lymphatic capillaries transport absorbed fats away from the intestine and deliver these molecules to the blood. Figure 4.17 Blood. A fluid connective tissue. Blood is a fluid connective tissue containing erythrocytes and various types of leukocytes that circulate in a liquid extracellular matrix. LM times 1600. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. View the University of Michigan webscope at http://virtualslides.med.umich.edu/histology/cardiovascular% 20system/081-3_histo_40x.svs/view.apml http://openstaxcollege.org/l/cardiovascular to explore the tissue sample in greater detail. 159. Visit this link. http://openstaxcollege.org/l/10 quiz to test your connective tissue knowledge with this 10-question quiz. Can you name the 10 tissue types shown in the histology slides? 4.4. Muscle tissue and motion. By the end of this section, you will be able to identify the 3 types of muscle tissue, compare and contrast the functions of each muscle tissue type, explain how muscle tissue can enable motion. Muscle tissue is characterized by properties that allow movement. Muscle cells are excitable. They respond to a stimulus. They are contractile, meaning they can shorten and generate a pulling force. When attached between two movable objects, in other words, bones, contractions of the muscles cause the bones to move. Some muscle movement is voluntary, which means it is under conscious control. For example, a person decides to open a book and read a chapter on anatomy. Other movements are involuntary, meaning they are not under conscious control, such as the contraction of your pupil in bright light. Muscle tissue is classified into 3 types according to structure and function: skeletal, cardiac, and smooth. Table 4.2. Comparison of structure and properties of muscle tissue types. Tissue histology. Function. Location: skeletal, long cylindrical fiber, striated, many peripherally located nuclei. Voluntary movement, produces heat, protects organs attached to bones and around entrance points to body, e.g., mouth, anus. Cardiac. Short, branched, striated, single central nucleus contracts to pump blood. Heart. Smooth, short, spindle-shaped, no evident striation, single nucleus in each fiber. Involuntary movement, moves food, involuntary control of respiration, moves secretions, regulates flow of blood in arteries by contraction. Walls of major organs and passageways. Table 4.2. Skeletal muscle is attached to bones and its contraction makes possible locomotion, facial expressions, posture, and other voluntary movements of the body. 40% of your body mass is made up of skeletal muscle. Skeletal muscles generate heat as a byproduct of their contraction and thus participate in thermal homeostasis. Shivering is an involuntary contraction of skeletal muscles in response to perceived lower than normal body temperature. The muscle cell, or myocyte, 161.8, develops from myoblasts derived from the mesoderm. Myocytes and their numbers remain relatively constant throughout life. Skeletal muscle tissue is arranged in bundles surrounded by connective tissue. Under the light microscope, muscle cells appear striated with many nuclei squeezed along the membranes. The striation is due to the regular alternation of the contractile proteins actin and myosin, along with the structural proteins that couple the contractile proteins to connective tissues. The cells are multinucleated as a result of the fusion of the many myoblasts that fuse to form each long muscle fiber. Cardiac muscle forms the contractile walls of the heart. The cells of cardiac muscle, known as cardiomyocytes, also appear striated under the microscope. Unlike skeletal muscle fibers, cardiomyocytes are single cells typically with a single centrally located nucleus. A principal characteristic of cardiomyocytes is that they contract on their own intrinsic rhythms without any external stimulation. Cardiomyocytes attach to one another with specialized cell junctions called intercalated discs. Intercalated discs have both anchoring junctions and gap junctions. Attached cells form long, branching cardiac muscle fibers that are, essentially, a mechanical and electrochemical syncytium allowing the cells to synchronize their actions. The cardiac muscle pumps blood through the body and is under involuntary control. The attachment junctions hold adjacent cells together across the dynamic pressures changes of the cardiac cycle. Smooth muscle tissue contraction is responsible for involuntary movements in the internal organs. It forms the contractile component of the digestive, urinary, and reproductive systems as well as the airways and arteries. Each cell is spindle-shaped with a single nucleus and no visible striations. Figure 4.18. Figure 4.18 Muscle tissue. Skeletal muscle cells have prominent striation and nuclei on their periphery. B. Smooth muscle cells have a single nucleus and no visible striations. C. Cardiac muscle cells appear striated and have a single nucleus. From top. LM times 1600. LM times 1600. LM times 1600. Micrographs provided by the Regents of University of Michigan Medical School. Copyright 2012. 161. Watch this video. http://openstaxcollege.org/l/musktissue. To learn more about muscle tissue. In looking through a microscope, how could you distinguish skeletal muscle tissue from smooth muscle? 4.5. Nervous tissue mediates perception and response. By the end of this section, you will be able to identify the classes of cells that make up nervous tissue. Discuss how nervous tissue mediates perception and response. Nervous tissue is characterized as being excitable and capable of sending and receiving electrochemical signals that provide the body with information. Two main classes of cells make up nervous tissue: the neuron and neuroglia. Figure 4.19. Neurons propagate information via electrochemical impulses, called action potentials, which are biochemically linked to the release of chemical signals. Neuroglia play an essential role in supporting neurons and modulating their information propagation. Figure 4.19. The neuron. The cell body of a neuron, also called the soma, contains the nucleus and mitochondria. The dendrites transfer the nerve impulse to the soma. The axon carries the action potential away to another excitable cell. LM times 1600. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. 162. 1.8. Follow this link. http://openstaxcollege.org/l/nobel. To learn more about nervous tissue. What are the main parts of a nerve cell? Neurons display distinctive morphology, well suited to their role as conducting cells, with three main parts. The cell body includes most of the cytoplasm, the organelles, and the nucleus. Dendrites branch off the cell body and appear as thin extensions. A long tail, the axon, extends from the neuron body and can be wrapped in an insulating layer known as myelin, which is formed by accessory cells. The synapse is the gap between nerve cells, or between a nerve cell and its target, for example, a muscle or a gland, across which the impulse is transmitted by chemical compounds known as neurotransmitters. Neurons categorized as multipolar neurons have several dendrites and a single prominent axon. Bipolar neurons possess a single dendrite and axon with the cell body, while unipolar neurons have only a single process extending out from the cell body, which divides into a functional dendrite and into a functional axon. When a neuron is sufficiently stimulated, it generates an action potential that propagates down the axon towards the synapse. If enough neurotransmitters are released at the synapse to stimulate the next neuron or target, a response is generated. The second class of neural cells comprises the neuroglia or glial cells, which have been characterized as having a simple support role. The word glia comes from the Greek word for glue. Recent research is shedding light on the more complex role of neuroglia in the function of the brain and nervous system. Astrocyte cells, named for their distinctive star shape, are abundant in the central nervous system. The astrocytes have many functions, including regulation of ion concentration in the intercellular space, uptake and/or breakdown of some neurotransmitters, and formation of the blood-brain barrier, the membrane, that separates the circulatory system from the brain. Microglia protect the nervous system against infection but are not nervous tissue because they are related to macrophages. Oligodendrocyte cells produce myelin in the central nervous system, brain and spinal cord, while the Schwann cell produces myelin in the peripheral nervous system. Figure 4.20. Figure 4.20 Nervous tissue. Nervous tissue is made up of neurons and neuroglia. The cells of nervous tissue are specialized to transmit and receive impulses. LM times 872. Micrograph provided by the Regents of University of Michigan Medical School. Copyright 2012. 163.4.6. Tissue injury and aging. By the end of this section, you will be able to identify the cardinal signs of inflammation, list the body's response to tissue injury, explain the process of tissue repair, discuss the progressive impact of aging on tissue, describe cancerous mutations' effect on tissue. Tissues of all types are vulnerable to injury and, inevitably, aging. In the former case, understanding how tissues respond to damage can guide strategies to aid repair. In the latter case, understanding the impact of aging can help in the search for ways to diminish its effects. Tissue injury and repair. Inflammation is the standard, initial response of the body to injury. Whether biological, chemical, physical, or radiation burns, all injuries lead to the same sequence of physiological events. Inflammation limits the extent of injury, partially or fully eliminates the cause of injury, and initiates repair and regeneration of damaged tissue. Necrosis, or accidental cell death, causes inflammation. Apoptosis is programmed cell death, a normal step-by-step process that destroys cells no longer needed by the body. By mechanisms still under investigation, apoptosis does not initiate the inflammatory response. Acute inflammation resolves over time by the healing of tissue. If inflammation persists, it becomes chronic and leads to diseased conditions. Arthritis and tuberculosis are examples of chronic inflammation. The suffix -itis denotes inflammation of a specific organ or type. For example, peritonitis is the inflammation of the peritoneum, and meningitis refers to the inflammation of the meninges, the tough membranes that surround the central nervous system. The four cardinal signs of inflammation, redness, swelling, pain, and local heat, were first recorded in antiquity. Cornelius Celsus is credited with documenting these signs during the days of the Roman Empire, as early as the 1st century AD. A fifth sign, loss of function, may also accompany inflammation. Upon tissue injury, damaged cells release inflammatory chemical signals that evoke local vasodilation, the widening of the blood vessels. Increased blood flow results in apparent redness and heat. In response to injury, mast cells present in tissue degranulate, releasing the potent vasodilator histamine. Increased blood flow and inflammatory mediators recruit white blood cells to the site of inflammation. The endothelium lining the local blood vessel becomes leaky under the influence of histamine and other inflammatory mediators, allowing neutrophils, macrophages, and fluid to move from the blood into the interstitial tissue spaces. The excess liquid in tissue causes swelling, more properly called edema. The swollen tissue's squeezing pain receptors cause the sensation of pain. Prostaglandins released from injured cells also activate pain neurons. Non-steroidal anti-inflammatory drugs, NSAIDs, reduce pain because they inhibit the synthesis of prostaglandins. High levels of NSAIDs reduce inflammation. Antihistamines decrease allergies by blocking histamine receptors and as a result the histamine response. After containment of an injury, the tissue repair phase starts with removal of toxins and waste products. Clotting, coagulation, reduces blood loss from damaged blood vessels and forms a network of fibrin proteins that trap blood cells and bind the edges of the wound together. A scab forms when the clot dries, reducing the risk of infection. Sometimes a mixture of dead leukocytes and fluid called pus accumulates in the wound. As healing progresses, fibroblasts from the surrounding connective tissues replace the collagen and extracellular material lost by the injury. Angiogenesis, the growth of new blood vessels, results in vascularization of the new tissue known as granulation tissue. The clot retracts, pulling the edges of the wound together, and it slowly dissolves as the tissue is repaired. When a large amount of granulation tissue forms and capillaries disappear, a pale scar is often visible in the healed area. A primary union describes the healing of a wound where the edges are close together. When there is a gaping wound, it takes longer to refill the area with cells and collagen. The process called secondary union occurs as the edges of the wound are pulled together by what is called wound contraction. When a wound is more than 1/4 of an inch deep, sutures, stitches, are recommended to promote a primary union and avoid the formation of a disfiguring scar. Regeneration is the addition of new cells of the same type as the ones that were injured. Figure 4.21. 164 1.8 Figure 4.21 Tissue healing during wound repair. Collagen fibers are laid down randomly by fibroblasts that move in to repair the area. Watch this video. http://openstaxcollege.org/l/healing hand to see a hand heal. Over what period of time do you think these images were taken? Tissue and aging. According to poet Ralph Waldo Emerson, the surest poison is time. In fact, biology confirms that many functions of the body decline with age. All the cells, tissues, and organs are affected by senescence, with noticeable variability between individuals owing to different genetic makeup and lifestyles. The outward signs of aging are easily recognizable. The skin and other tissues become thinner and drier, reducing their elasticity, contributing to wrinkles and high blood pressure. Hair turns gray because follicles produce less melanin, the brown pigment of hair and the iris of the eye. The face looks flabby because elastic and collagen fibers decrease in connective tissue and muscle tone is lost. Glasses and hearing aids may become parts of life as the senses slowly deteriorate, all due to reduced elasticity. Overall height decreases as the bones lose calcium and other minerals. With age, fluid decreases in the fibrous cartilage discs intercalated between the vertebrae in the spine. Joints lose cartilage and stiffen. Many tissues, including those in muscles, lose mass through a process called atrophy. Lumps and rigidity become more widespread. As a consequence, the passageways, blood vessels, and airways become more rigid. The brain and spinal cord lose mass. Nerves do not transmit impulses with the same speed and frequency as in the past. Some loss of thought clarity and memory can accompany aging. More severe problems are not necessarily associated with the aging process and may be symptoms of underlying illness. As exterior signs of aging increase, so do the interior signs, which are not as noticeable. The incidence of heart diseases, respiratory syndromes, and type 2 diabetes increases with age, though these are not necessarily age-dependent effects. Wound healing is slower in the elderly, accompanied by a higher frequency of infection as the capacity of the immune system to fend off pathogen declines. Aging is also apparent at the cellular level because all cells experience changes with aging. Telomeres, regions of the chromosomes necessary for cell division, shorten each time cells divide. As they do, cells are less able to divide and 165 regenerate. Because of alterations in cell membranes, transport of oxygen and nutrients into the cell and removal of carbon dioxide and waste products from the cell are not as efficient in the elderly. Cells may begin to function abnormally, which may lead to diseases associated with aging, including arthritis, memory issues, and some cancers. The progressive impact of aging on the body varies considerably among individuals, but studies indicate, however, that exercise and healthy lifestyle choices can slow down the deterioration of the body that comes with old age. 166 1.8 Tissues and cancer. Cancer is a generic term for many diseases in which cells escape regulatory signals. Uncontrolled growth, invasion into adjacent tissues, and colonization of other organs, if not treated early enough, are its hallmarks. Health suffers when tumors rob blood supply from the normal organs. A mutation is defined as a permanent change in the DNA of a cell. Epigenetic modifications, changes that do not affect the code of the DNA but alter how the DNA is decoded, are also known to generate abnormal cells. Alterations in the genetic material may be caused by environmental agents, infectious agents, or errors in the replication of DNA that accumulate with age. Many mutations do not cause any noticeable change in the functions of a cell. However, if the modification affects key proteins that have an impact on the cell's ability to proliferate in an orderly fashion, the cell starts to divide abnormally. As changes in cells accumulate, they lose their ability to form regular tissues. A tumor, a mass of cells displaying abnormal architecture, forms in the tissue. Many tumors are benign, meaning they do not metastasize nor cause disease. A tumor becomes malignant, or cancerous, when it breaches the confines of its tissue, promotes angiogenesis, attracts the growth of capillaries, and metastasizes to other organs. Figure 4.22. The specific names of cancers reflect the tissue of origin. Cancers derived from epithelial cells are referred to as carcinomas. Cancer in myeloid tissue or blood cells form myelomas. Leukemias are cancers of white blood cells, whereas sarcomas derive from connective tissue. Cells in tumors differ both in structure and function. Some cells, called cancer stem cells, appear to be a subtype of cell responsible for uncontrolled growth. Recent research shows that contrary to what was previously assumed, tumors are not disorganized masses of cells, but have their own structures. 167 Figure 4.22 Development of cancer. Note the change in cell size, nucleus size, and organization in the tissue. 168 1.8 Watch this video. http://openstaxcollege.org/l/tumor to learn more about tumors. What is a tumor? Cancer treatments vary depending on the disease's type and stage. Traditional approaches, including surgery, radiation, chemotherapy, and hormonal therapy, aim to remove or kill rapidly dividing cancer cells, but these strategies have their limitations. Depending on a tumor's location, for example, cancer surgeons may be unable to remove it. Radiation and chemotherapy are difficult, and it is often impossible to target only the cancer cells. The treatments inevitably destroy healthy tissue as well. To address this, researchers are working on pharmaceuticals that can target specific proteins implicated in cancer-associated molecular pathways.